Yeah, welcome back to the channel, and today we focus on blood groups. Our focus here is simply how blood groups are inherited in a family, right? So let's get started. With blood groups, the main point is that there are four main blood groups. That is, you can either have blood group A, blood group B, blood group AB, or blood group O. So there are only four blood groups that are possible. It is blood group A, blood group B, blood group AB, and finally blood group O. Now, these blood groups will be inherited in a family, and we'll have to show that through a genetic cross, and we'll do that at a later stage. But the starting point here is that there's a grand total of just four blood groups, right? Let's continue. Now, although we have the four blood groups, these blood groups are controlled by one gene. Not only are they controlled by one gene, but they're also controlled by three alleles. Now, this is a very simple statement to understand, in the sense that we know that each and every single characteristic within our body, it is going to be controlled by one gene, and it is going to be the gene for that particular trait. Now, with blood groups, it is going to be one gene, because this is the gene for blood groups, right? So it is going to be one gene. But there's a grand total of three alleles that control these blood groups. So even though a person can only inherit one blood group at a time, and also you are going to inherit two alleles at a time, because each and every characteristic is controlled by two alleles. It is going to be an allele from the paternal origin and an allele from the maternal origin. So you can only have two at a time. But in total, there's three alleles that control blood groups, right? Let's try to look at the alleles that are responsible for blood groups. Number one, we've got the allele capital letter I to the exponent of capital letter A. Now, this is how we write alleles with respect to blood groups. We don't just write A or capital letter I. We write capital letter I to the exponent of capital letter A. Now, this is the allele for blood group A. Now, you will note that when we write this allele, we're using capital letters. And the reason why we are using capital letters is that we're trying to show that this allele is dominant, right? We also have another allele here, which is the allele for blood group B. The allele for blood group B is written as capital letter I to the exponent of capital letter B. So this is the allele for blood group B. You will also note that when we write this allele, we're using capital letters. And the reason why we use capital letters to denote alleles that are dominant. So here we can see that the allele for blood group A is written by capital letter I to the exponent of capital letter A, telling us that it is dominant. The same applies to the allele for blood group B, which is written as capital letter I to the exponent of capital letter B, telling us that it is also dominant. And finally, we've got small letter I. Small letter I here is the allele that controls blood group O. But since we are writing the allele for blood group O as small letter I, it tells us that it is recessive, right? So we've got three alleles in total. We've got capital letter I, capital letter A, that is the allele for blood group A. We've got capital letter I to the exponent of capital letter B, that is the allele for blood group B. And finally, we've got small letter I, which is the allele for blood group O. What we can note here is that the allele for blood group O, which is small letter I, is recessive to the allele for blood group A. It is also recessive to the allele for blood group B, right? So the allele for blood group A is dominant over the allele for blood group O. The allele for blood group B is also dominant over the allele for blood group O. But the alleles for blood group A and the alleles for blood group O are what we call codominant. Remember, the idea of codominance is that the alleles are equally dominant. And when these alleles are equally dominant, it tells us that in a heterozygous condition, they are going to be expressed equally in the phenotype. So as such, that is how we have capital letter I to the exponent of capital letter A, capital letter I to the exponent of capital letter B. And this will give us blood group AB. So the inheritance of blood group AB is a sign or is an example of codominance, simply because the allele for blood group A and the allele for blood group B are codominant. They are equally dominant, and when they are equally dominant, they are expressed equally in the phenotype, right? But the allele for blood group O, as we've already said, is going to be recessive to the alleles for blood group E and blood group B, respectively. Let us continue. Now, we know that we've got these 4 blood groups, and the blood groups here are going to be what we call the phenotype. So when we describe the actual blood group, for example, when I say blood group A, that is going to be the phenotype for blood group A. We simply call it blood group A. The same applies to blood group B. That is the phenotype for blood group B, and so on with blood group AB and blood group O, right? But let's try to look at the possible genotypes for a person who has got blood group A. Let's look at the possible genotypes. First of all, a person with blood group A can be homozygous. In this sense, we're going to say this is homozygous dominant, right? So a person with blood group A can inherit capital letter I to the exponent of capital letter A from the paternal origin, or capital letter I, capital letter A from the maternal origin. In this sense, this person is going to have blood group A. But a person with blood group A can also be heterozygous. Remember, the word hetero will simply mean different. The word homo will simply mean same, right? So if a person, for example, inherits capital letter I to the exponent of capital letter A from the paternal origin and small letter I from the maternal origin, this person is going to have blood group A. And that is simply because we know that the allele for blood group A is dominant to the allele for blood group O. So in this example, the allele for blood group A is going to determine the phenotype. Hence, we are saying that this person is going to have blood group A. And the effects of small letter I are going to be masked. So this person is going to have blood group A. And this is a sign of complete dominance, because in this heterozygous state, the dominant allele here determines the phenotype, right? Let's go to blood group B. The same is also applied to blood group B. A person with blood group B can be homozygous. This person can be homozygous, remembering that the word homo in this instance will simply mean same, right? So capital letter I to the exponent of capital letter A can be from the paternal origin, and capital letter I, capital letter B can be from the maternal origin, and this will result in an individual with blood group B. But a person with blood group B can also be heterozygous. This is also possible. This person can also be heterozygous, remembering that the word hetero in this instance will simply mean different. So capital letter I to the exponent of capital letter B will be inherited from the paternal origin, and small letter I can be inherited from the maternal origin. So in this heterozygous state, this person is going to have blood group B, and that is simply because we know that the allele for blood group B is dominant to the allele for blood group O. As such, the effects of small letter I here are going to be masked, and capital letter I to the exponent of capital letter B is going to determine the phenotype. As such, this is also a type of complete dominance. So this person will have blood group B. Let's go to blood group AB. Now, with blood group AB, there is only one possible genotype. Remember, with blood group AB, we said that the inheritance of blood group AB is a sign or is an example of codominance. It is an example of codominance. And in codominance, we said that the alleles are equally dominant. When they're equally dominant, they are expressed equally in the phenotype, right? So in order for a person to have blood group AB, this person must inherit capital letter I to the exponent of capital letter A from the paternal origin, and capital letter I to the exponent of capital letter B from the maternal origin. This can be vice versa. It can be from the maternal and paternal, or paternal and maternal. It doesn't really matter. So long as the two alleles interact, this is how we're going to write the genotype, and this person will have blood group AB. Again, the inheritance of blood group AB is a sign of what? It is a sign of codominance, and in codominance, the alleles are equally dominant. When they're equally dominant, they're going to be equally expressed in the phenotype. So allele, capital letter I to the exponent of capital letter A, capital letter I to the exponent of capital letter B will give us blood group AB. And then finally, with blood group O, there's only one possible genotype here as well. Now, since we said that the allele for blood group O is recessive, we said that the allele for blood group O is recessive to the allele for blood group A, and it is also recessive to the allele for blood group B, we know that in order for the recessive trait to be expressed in the phenotype, the genotype must be made of two recessive alleles, and this is also the case with blood group O. Since the allele for blood group O is small letter I, it tells us that small letter I is recessive. And in order for small letter I to determine the phenotype, the genotype must be small letter I, and therefore this will give us our blood group O. If this was in a heterozygous condition, the effects of small letter I were going to be masked since it is recessive, right? So these are our 4 blood groups, and these are the possible genotypes that are needed in order to inherit one of those blood groups. Let us continue. Now let's look at the inheritance of alleles where blood groups are concerned. Now we've already stated that we have got a grand total of 3 alleles that control blood groups. But even if there are 3 alleles controlling blood groups, an individual can only inherit 2 of these alleles. It is impossible for any one individual to inherit all 3. And the reason why it is impossible is that each and every single characteristic is going to be controlled by 2 alleles. And one allele is from the paternal origin, the other allele is from the maternal origin, right? So you can only inherit one allele from each parent. So as such, there are only 2 alleles that an individual can inherit. Although the blood groups as a whole are controlled by 3 alleles, an individual can only get 2 at a time. Let's continue. Now let's do examples that involve blood groups. Here it says that a man with blood group AB. Let's look at this. We know that the inheritance of blood group AB is an example of codominance. The alleles for blood group A and the allele for blood group B are equally dominant. So we know that the genotype of these men is going to be capital letter I to the exponent of capital letter A, capital letter I to the exponent of capital letter B. That is the only possible genotype for a person with blood group AB, right? And a woman who is heterozygous for blood group B. Now, they were so kind to tell us that this woman is going to be heterozygous. We know that the word hetero in this instance will simply mean different, simply meaning that our genotype is going to be made up of different alleles. So, for this person to have blood group B and also be heterozygous, it tells us that the only possible genotype is going to be capital letter I to the exponent of capital letter B, small letter I. Now, that is going to be heterozygous. If this person was homozygous, it was going to be capital letter I to the exponent of capital letter B, capital letter I to the exponent of capital letter B. But because the person is said to be heterozygous, this is going to be our genotype, right? So these men and these women are said to be planning to have children. Let's try to answer the questions. 9.1. How many alleles control the inheritance of blood groups? We know there is a grand total of 3 alleles that control the inheritance of blood groups, right? 9.2. Describe the type of dominance that occurs in the inheritance of blood group B in the woman. Let's look at blood group B. In this woman, the woman is said to be heterozygous. So when this woman is heterozygous, she has got an allele for blood group B and she also have an allele for blood group O, which is small letter I. But irrespective of the presence of the allele for blood group O, it has been completely masked since the woman is said to have blood group B, and this is an example of what we call complete dominance. This is what we call complete dominance, right? Why is it complete dominance? It is complete dominance because the allele for blood group B, which is capital letter I to the exponent of capital letter B here, is dominant to the allele for blood group O, which is small letter I. As such, in a heterozygous condition, the dominant allele, which is the allele for blood group B, determines the phenotype. Hence, the woman has blood group B. So the allele for blood group O here has been completely dominated. So this is complete dominance. Right? 9.3. They say that use a genetic cross to show all the possible genotypes and phenotypes of their children. So we're going to do a genetic cross to show the possible genotypes and phenotypes of these parents. So it is a man with blood group AB and a woman who is heterozygous for blood group B. Let us make that genetic cross. Now we know that our genetic cross file left, we start with P1, and that simply stands for parents of the first generation. In front of P1, we have to write phenotype. And we know that phenotype simply means the physical appearance of an organism. But we know that with blood groups, the actual blood group itself is the phenotype. So in this sense, we're going to write blood group AB. This is the man. The man is said to have blood group AB. So this man is going to be crossed with the woman who is said to have blood group B. So we're going to write blood group B. Blood group B. We're going to write blood group B, right? Let us continue. Below phenotype, we're going to write genotype. Now, there is only one possible genotype for an individual who is said to have blood group AB, and that possible genotype is capital letter I to the exponent of capital letter A, capital letter I to the exponent of capital letter B, cross. This woman was said to be heterozygous for blood group B, which means that a genotype is capital letter I, capital letter B, and small letter I. That is the woman's genotype, right? Below genotype, we're going to write meiosis. And in front of meiosis, we write nothing. So let us continue. Below meiosis, now comes gametes. When we write our gametes, we simply separate the genotype by a comma. So it is going to be capital letter I, capital letter A, comma, capital letter I, capital letter B, crossed with capital letter I, capital letter B, comma, small letter I. Below gametes now comes fertilization. And we're going to show this fertilization by means of a Punnett square. So with a Punnett square, that is 3 boxes to the side and 3 boxes downwards. So it is going to be 3 boxes to the side and 3 boxes downwards. In the top left box, we're going to simply write gametes. Now in this box and that box, we're simply going to write the gametes of parent number 1. And the first gamete here was capital letter I to the exponent of capital letter A. The second gamete was capital letter I to the exponent of capital letter B. In the boxes below the word gametes, we're going to write the gametes of parent number 2. And the first gamete for parent number 2 there was capital letter I to the exponent of capital letter B, followed by small letter I, right? Now in this box, we're going to cross the allele for blood group A and the allele for blood group B. So it is going to be capital letter I, capital letter A, capital letter I, capital letter B. In this box, we're going to cross the allele for blood group B and the allele for blood group B. So it is going to be capital letter I to the exponent of capital letter B, capital letter I to the exponent of capital letter B. In this box, it is going to be the allele for blood group A and the allele for blood group O. So here it is going to be capital letter I to the exponent of capital letter A, small letter I. In this box, it is going to be the allele for blood group B and the allele for blood group O. So it is going to be capital letter I, capital letter B, small letter I. That is the genotype of their offspring, right? So below fertilization now comes F1. F1 simply meaning offspring of the first generation. In front of the word F1, we're going to write genotype. We're going to simply copy the genotype of the offspring there. So it is going to be capital letter I, capital letter A, capital letter I, capital letter B, capital letter I, capital letter E, small letter I, capital letter I, capital letter B, capital letter I, capital letter B, capital letter I, capital letter B, and small letter I. That is the genotype of the offspring. We simply copy them from our Punnett square. Below genotype now comes phenotype. Now comes phenotype. Now let's look at the phenotype of the offspring. So we know that this offspring is going to have blood group AB, right? This offspring is going to have blood group A. That is simply because the allele for blood group A is dominant to the allele for blood group O. And this person is going to be homozygous for blood group B, and this person is going to be heterozygous for blood group B. Now let's write the phenotype. So we're going to have one blood group AB, right? We're also going to have one blood group A. We're going to have one blood group A. And then finally, we're going to have two blood group B. We're going to have two blood group B. And that is our genetic cross done. Let's continue again and do another example. Example 2 here says that human blood groups are controlled by multiple alleles. Yes, we know it is multiple alleles. It is 3 of them, right? 3.2.1, how many alleles control blood groups? We know the answer, it is 3. 3.2.2, which two alleles are codominants in the inheritance of blood groups? We know the alleles that are codominant. It is the allele for blood group A and the allele for blood group B. These are the alleles that are codominant, right? 3.2.3, a man is heterozygous for blood group A. So if this man is said to be heterozygous, we know that the genotype for this man can only be I capital letter A small letter I. That is the only possible genotype for this man who is said to be heterozygous, right? And marries a woman who has blood group O. Now we know that the allele for blood group O is recessive. And in order for the recessive allele to determine the phenotype, the genotype must be made of two recessive alleles. So in order for this individual to have blood group O, the only possible genotype for this individual is small letter I, right? So use a genetic cross to show the phenotypic ratio of their offspring. We're required to show the phenotypic ratio. So it is a man who is heterozygous for blood group A and a woman who has blood group O. Let us perform that genetic cross. Again, top left, we're simply going to write P1. In front of P1, we're going to write phenotype. That is the physical appearance. So what is our phenotype? They said it was a man who was heterozygous for blood group A. It is a man who was heterozygous for blood group A. They said he married a woman who had blood group O. They said that man married a woman who had blood group O. So that is their phenotype. That is the physical appearance, right? Below phenotype now comes genotype. Now the man was said to be heterozygous, telling us that his genotype can only be capital letter A, capital letter I to the exponent of capital letter A, and small letter I, crossed with the woman with blood group O. Blood group O can only be small letter I, small letter I. Below genotype comes what? Comes meiosis. In front of meiosis, we write nothing. Below meiosis now comes gametes. With gametes, we simply separate the genotype by comma. So it is going to be capital letter AI to the exponent of capital letter A, comma, small letter I, crossed with small letter I, comma, small letter I. Below gametes now comes fertilization. And we show fertilization by means of a Punnett square. 3 boxes sideways, 3 boxes downwards. 1, 2, 3. In the box that is top left, we write gametes again. In front of the word gametes, we're simply going to write the gametes of the first parent here, starting with the first gamete, which is capital letter I to the exponent of capital letter A, followed by small letter I. Below the word gametes, we're going to write the gametes of parent number 2 there, starting with small letter I, small letter I. In this box, we're going to cross the allele for blood group A and the allele for blood group O, so it is going to be capital letter I, capital letter A, small letter I. In this box, we're going to cross the allele for blood group O and the allele for blood group O. So the genotype here is going to be small letter I, small letter I. In this box, we're going to cross the allele for blood group A and the allele for blood group O. So it is going to be capital letter I to the exponent of capital letter A, small letter I. And in this box, we're going to cross the allele for blood group O and the allele for blood group O. So it is going to be small letter I, small letter I. Below fertilization now comes F1. These are offspring of the first generation. In front of F1, we simply write genotype. And we're going to copy our genotype as is. So it is going to be capital letter I to the exponent of capital letter A, small letter I, capital letter I to the exponent of capital letter A, small letter I, small letter I, small letter I, small letter I, small letter I. That is our genotype. Below genotype now comes phenotype. Right? So let's look at this individual. This individual is heterozygous for blood group A because we know that the allele for blood group A is dominant over the allele for blood group O. So this person has blood group A. Same with this one. So these two are both blood group A. With this one, the genotype is small letter I, and these are recessive alleles. And we know that small letter I is the allele for blood group O. And since it is recessive, it can only influence the genotype if the genotype is made up of two small letters. So this person is going to have blood group O. Same with the last offspring. So these two are blood group O and these two are blood group A, right? So our phenotype here is going to be 2 blood group A. It is going to be 2 blood group A and 2 blood group O. But we have not answered the question. The question wanted us to write the phenotypic ratio. So we're going to write phenotypic ratio. So we've got 2 that are blood group A and 2 that are blood group O. But it is very important to note when writing the phenotypic ratio or the genotypic ratio, it must always be written in simplest form, right? So we can simplify 2:2 in the sense that 2 goes into 2 once and 2 will go into 2 once. So our final answer here is going to be 1:1. That is going to be our final answer, right? And that is our genetic cross done. Let us continue. Now let's briefly look at the use of blood group in paternity testing. This is where we want to determine if a particular man is the father to a particular child, right? But when we use blood group in paternity testing, it is important to note that we can only use blood groups to exclude a particular man as the parent. So we can only use these blood groups to exclude, to say that this man is not the father to this child. But we cannot use blood groups to confirm that this particular man is the father. I repeat, when we use blood groups in paternity testing, we can only use these blood groups to exclude a particular man as the father. But the use of blood groups cannot confirm that this particular man is the father to this child. It is impossible. Why is it impossible? It is simply because many people around the world share the same blood groups, right? Many people around the world share the same blood group. So it is possible for multiple men to have a child with the same blood group that matches yours with the very same woman, for example, right? Let us make an example that is a real world example. Let's say you've got a woman and you suspect the woman of infidelity, you suspect her of cheating, right? And let's say the woman has blood group B, for example, and you as the man, you have blood group O. Let's put it this way, right? And the woman that the child that the woman gives birth to, let's say, has got blood group AB, right? We can use these blood groups to exclude this man as the father to this child, as it is impossible for this father to have a child with blood group AB. This is simply because a child that has got blood group AB must have inherited the allele for blood group A from one of the parent. The same with the allele for blood group B, which the man simply doesn't have. So we can use blood groups to exclude this man as the father of this child, right? Let's look at another example. Let's say it is the very same man with blood group O and the very same woman with blood group B. And this couple gives birth to a child that has got blood group B. Let's look at this cross. So it is possible for these two parents now to produce a child that has got blood group B. That is simply because this child will receive the allele for blood group B from the mother and will receive the recessive allele for blood group O from the father. So this child can be heterozygous for blood group B. It makes sense that this man can be the father, but we cannot be conclusive and say that this man is the father. This is simply because there is another man out there in the world with the very same blood group O. And as such, if this man was to be crossed with your wife, they would also have a child with blood group B, right? So because many people share the same blood groups, we cannot use blood groups to confirm that a particular man is the father to a child. In order to be conclusive and confirm that a particular man is the father to the child, we can only use DNA profiling. With blood groups, we can only exclude the man as the father. We cannot confirm him to be the father of that particular child, right? And this is how blood groups are used in paternity testing. If you like what I have had to say, please consider liking and subscribing to the channel. Until next time, bye-bye.